Hey all. So I am recording this video at school on Pancake, and so who knows what might happen while I am talking. All heck could break loose. So in recording this video, we are going to be looking at A2.3, which is viruses, and we're going to try to answer the questions: how can viruses exist with so few genes, and in what ways do viruses vary? So structural features of viruses are quite different. And the way that they work is they're non-living. So they have DNA or RNA inside them. They infect the host and they hijack the functions to get the cell to make more of the virus. There are multiple origins probably of viruses, meaning they originated multiple times because they share so few features. They're very small and they don't increase in size. They are assembled in a host cell and they use different nucleic acids as genetic material. So some viruses have RNA and some have DNA. And that allows them to use translation in the host cells. They have capsids made of proteins, and that's going to hold the genetic material. But where the capsid is can differ as well. Capsids are very symmetrical. And once they're assembled, they have no cytoplasm and very few enzymes, but they do use enzymes in many cases to replicate DNA or RNA for infecting the host. So the evidence of several origin locations for viruses could be, so one theory hypothesis related to viruses is that they were obligate parasites, meaning they need cells. So they must have evolved, cells must have evolved first, and then viruses came along after, because otherwise they wouldn't have had a host to make their protein, their genetic material. They use the same genetic code as cells, so they could have originally come from cells and then changed. So there's a progressive hypothesis where viruses moved step by step from cells. Eukaryotes contain transposons, and transposons make DNA from RNA. And that's similar to how HIV replicates. So if viruses also contain transposons and they are able to get cells to replicate for them, maybe they used to be cells and then they've changed. And then the regressive hypothesis is that they were a parasitic bacteria that gradually lost their life functions and outsourced it to host cells. So this is more like how chlamydia would function. But it could be both ways. So there's distinctive characteristics for viruses. They can have DNA or RNA, different lengths, single or double-stranded, circular or linear. They can replicate differently. So the sense strand RNA can act as messenger RNA, but antisense RNA will transcribe to make RNA. In the case of retroviruses, they use RNA to make DNA and then make messenger RNA. So there's a lot of different ways that viruses get their DNA made. A lot of them have an envelope versus a non-envelope. And when lysis occurs, some viruses get covered in phospholipid membranes from the host cells. This is usually for animals, and that allows the virus to bind with new cells. So plant or bacteria viruses don't typically have an envelope, and then they don't typically get covered in the phospholipid membrane to allow them to get into the different cells that they're trying to penetrate. So when we're talking about cells, we're looking at the viruses, we're looking at the lysogenic cycle. The lysogenic cycle, there's two stages of this. And so the first stage is this part where DNA is inserted into an organism, perhaps a bacteria, from a virus. So DNA is integrated into the bacterial DNA. Whole new particles are not produced unless triggered into the lytic stage. And so in the lytic stage, new viruses are produced, but in this stage, the lysogenic stage, just DNA is integrated into the cell from the virus. And this can actually cause quite a lot of mutations. And then if the lytic cycle is triggered, then the cell is going to burst and it's going to produce more viruses. So virulence is a problem for viruses. If all the host cells are killed, then the virus will die. So animals are fighting viruses with antibodies. And it's important to notice that there are two stages of this lysogenic cycle. So back to the types of different viruses. So this is in your textbook specifically. So bacteriophage lambda, bacteriophages work on bacteria. So they're viruses that infect bacteria. Their hosts are going to be bacteria or archaea. They don't have an envelope. They have one double strand of DNA, and they can be lytic or lysogenic. COVID, as an example, obviously their host is humans and mammals. It has an envelope and it has one strand of RNA. And this is zoonotic, meaning it was transmitted from animals to humans. HIV, the host is human T helper cells. It's a retrovirus, so that means it converts RNA to DNA and then back to messenger RNA. It has an envelope. It has 2 copies of RNA, which will make reverse transcriptase to copy that RNA to DNA. And HIV can also merge with other strains of HIV, and that will make even greater variation in that virus, which means you could be infected with 2 strains of HIV, and both of them would have different impacts on your immune system. So evolution can happen very quickly for viruses because their generation time is only about 1 hour. And mutations are very high in viruses, especially RNA viruses, because there's no checks in this replication process. So there's very strong selection pressures going on with viruses. So antigens can change. The immune system can then not attack with the antibodies. So viruses changing their antigens can be really useful for the virus success. And so, for example, one of the antigens on the surface of a virus would be hemagglutinin that allows it to bind to the cell, whereas neuroamidase allows it to release from the cell. So in the case of the flu virus, the RNA virus, this has a very high mutation rate. It has 8 RNA strands, and they can merge with other versions of the virus, and it can be transmitted between species. So when you're getting a flu vaccine, it typically has multiple versions of different strains of flu, like H1N1 versus H3N2. So these are all different strains of flu virus. Some of those are bird flu, some of those are swine flu, and then some of those are human flus. And flu viruses can also be zoonotic. HIV has the highest mutation rate of any virus. It can change the proteins on the surface, allowing new body cells to be hosts and become resistant to antiretroviral drugs. So HIV is really intense, and that's why we don't have a cure for HIV at this point. So to summarize, viruses are non-living because they require a host. They have many structural differences because they evolved independently multiple times. They can be in a lytic or a lysogenic stage, and virulence in viruses is problematic for their continuation. So they want to keep you alive, but they also want to transmit. So, but if they kill you off, then you're not going to be able to be a host for them. And they evolve quickly because there's a lot of mutation and strong selection pressure going on in this process. And so I managed to dodge all of the pancreatic things. I don't know how that happened, but yay me.